So every 2 years in the pastel deserts of Tucson, Arizona, there's a gathering of about 600 or so people who are really interested in understanding one of the hardest and most interesting problems, which is what is the basis of consciousness. And one kind of very unique thing about this conference is that it brings together people from many different traditions. So scientists, philosophers, spiritual seekers, therapists. And so it's kind of a very interesting venue because people who have very vastly different structures from which they're coming to study this problem come together and try to figure out a solution. And Stuart has kind of a very key role in developing this conference, and it really reflects his own kind of approach to studying consciousness as well. So he started becoming really interested in consciousness in the 60s, and soon thereafter went into medical school where he studied cancer. And one kind of very interesting thing about his studies with cancer cells is that he realized that the seemingly trivial operation of a cell separating into two cells actually requires great finesse. Somehow the genes that are tugged apart and separated into two cells land there with a great precision, which, while we take for granted, is something that goes horribly awry in things like cancer. And it seems like microtubules play a very key role in achieving this very precise separation. And around that time, Stuart found out that these microtubules also play a very key role in structuring the very complex dendritic and axonal structure of neurons. And looking at the way that these microtubule subunits tessellate with each other struck Stuart because it reminded him of computer matrices. And that actually started many decade-long obsession for Stuart in this realm, and slowly he started piecing together his ideas of this very beautiful structure that is pervasive throughout neurons, and tried to come up with ideas of what it might be able to achieve for neurons. And at that time started reading some of the stuff by Penrose, and Stuart and Roger were great complements to each other because whereas Roger had this beautiful kind of mechanism for computation that didn't have a structure, Stuart had the structure in mind that didn't have a mechanism, and so it started a very fruitful collaboration. And so here you'll hear some of the ideas that Stuart's been developing for decades, and interestingly enough, in many neuroscience departments, all of this stuff is very foreign. So for example, very few of us have actually picked up any of the ideas that Stuart will be presenting today because we're very firmly kind of wedged in this idea that spikes somehow mediate some and are sufficient for explaining all of this very profound experience that we're all having. So we really look forward to hearing what Stuart has to say. Thank you, Stuart.
Thank you, Gotham. Thank you all for being here. Thank you. Great turnout. So I'm a bit of a heretic being kind of outside the main field of neuroscience, but I followed it very closely. And my topic, quantum cognition and brain microtubules. And what you see here is a neuron. There's the nucleus. And the yellow are the microtubules, and red are actin. And basically, the key point is that most everybody looks at what's happening at the membrane and ignores what's going on inside. So in medicine, in my field, that'd be like being a dermatologist and forgetting about everything else. So let me begin by picking, taking issue with some conventional wisdom in cognitive neuroscience. These are kind of basic tenets of cognitive neuroscience that I've observed that I think we have to challenge. So the first is that consciousness emerges from complex synaptic interactions among many simple neurons. The second is that relevant neuronal information is completely membrane-based, based on the Hodgkin-Huxley neuron. That synaptic strengths mediate memory and learning, synaptic plasticity, Hebbian learning, LTP, and that classical Bayesian probability mathematics governs cognition. And this challenge is a fairly new one. So the bottom line and solution to all these issues is to consider a finer scale of information processing in the neuronal cytoskeleton. So I'm giving away the punchline here is that I think we need to go down to this level to find the roots of cognition and consciousness. So let's look at the first one. Cognition emerges from complex synaptic interaction among many simple neurons. So here's a biological neuron. Here's what computer scientists view as a neuron. And here's kind of a toy neuron. And basically integrate and fire. So the dendrite and cell body integrate inputs, usually thought of only at the membrane, but I will challenge that because I think it's happening inside also, to a threshold at the axon initiation segment to fire an output or spike. And generally, the dogma is that the spikes are the currency of the brain, of cognition and consciousness, although local field potentials, EEG, dendritic synchrony, come from the other side. I would say that really consciousness and the important aspects of cognition are happening in the dendrite, and the firing is just conveying the information from that to the next synapse, the next layer. So if we make a network of, if we make a network of these toy neurons at the bottom, like this, and put inputs at the left and have outputs, and this is like a poor man's cheap simulation of a neural network, you can see that we get simple neurons either firing or not firing, and we get different outputs, and depending with synaptic plasticity adjustment, we can get learning. The basic idea is that cognition stems from complex computation among simple neurons, spiking. However, when you think about it, single-cell organisms perform fairly complex cognitive functions. So here's a paramecium. One cell doesn't have any synapses. It bounces into an object and goes a different way. It avoids predators. It avoids obstacles, as you can see. It finds food. It finds a mate. It has sex with a partner. Over here is an X-rated picture of two paramecium coupling. And interestingly, the only time they're absolutely still is during sex. So there they are. And how do they do that? Well, they move around and they sense by these hair-like projections called cilia, which connect to internal cytoskeleton. The cilia are made of microtubules, 9 couplets of microtubules shown here, that are found in virtually all animal cells. We have them in our lungs, we have them in our GI tract, we have them all over the place. And they bend back and forth, and they also sense. So and then they go into an internal cytoskeleton, the same microtubules inside the cell. And Gautam mentioned, I don't know if you can see this, Gautam mentioned mitosis. So here's a cell dividing, and the yellow are the centrioles, which are basically like cilia. The blue are the chromosomes, and the red would be the microtubules pulling them apart. And what he was saying is that if this doesn't work perfectly, if you have an asymmetric mitosis, you get an abnormal genotype. And this was an old theory of cancer that goes back 100 years to Bovary and was picked up by Duisberg, who I think is here, that aneuploidy or abnormal mitosis causes cancer. Then the geneticists came in, and oncogenes and repressor genes. And to my mind, that really hasn't solved the problem, and people are going back to the idea that maybe mitosis per se might be the problem. But that's kind of a digression. Except that the cilia and centrioles are these structures here, and they're made up of, in this case, triplets. It can be doublets or triplets of microtubules in this megacylinder of 9 pairs or triplets of microtubules that form centrioles and their same structure of cilia. And in simple organisms, they sense photons. Euglena, the centrioles in Euglena, Albrecht Buehler showed that will detect light and point the cell in another direction. And the same structure is found in all our rod and cone cells. So every photon that gets into our eye has to go through the narrow stalk to get to the rhodopsin in the back. And through this structure, which are, and all primitive visual systems are ciliated ectoderm, have this sort of structure. And it's interesting that the structure is pretty much the same diameter as the wavelength of photons, suggesting that they might be some kind of optical devices or quantum optical devices capturing orbital or angular momentum or something. That's another digression, though. So let's get back to neurons. So in neurons, as we said, there are many microtubules inside neurons. This is a cartoon version of a neuron. And a couple things to notice. So here's the dendrite and the soma here. The microtubules are interrupted. They're not continuous, and they're in opposite direction. They have a polarity. And they're of mixed polarity here, interconnected by these microtubule-associated proteins. In axons, they're continuous and unipolar. So I think all the interesting processing is going on here, and you can see the blow-up of the microtubules there that form these networks that are anti-parallel networks, and also gap junctions in which, through which I think there can be information exchange from microtubule to microtubule, possibly by quantum entanglement or some other mechanism. So we get kind of a layer of integration here that can do collective integration, which would be much more efficient than individual integrate and fire. So we have kind of a collective integration possibility here going on in the dendrites and the cell bodies. Okay, moving on to the next conventional wisdom is that relevant neuronal information is completely membrane-based, the Hodgkin-Huxley neuron. So here you have the cell body and dendrites, as I said. Inputs come in, membrane potentials, integrate to a threshold here, and then firing or not firing in a digital binary mechanism onto the next layer. And that's kind of the party line. However, the firing threshold in brain neurons vary on a spike-to-spike basis due to non-membrane factors, as shown in this paper by Nondorf et al. And others in 2007. So here's what the Hodgkin-Huxley neuron would predict, that you integrate to a threshold, a very narrow threshold, and you fire within a very narrow temporal range. However, actual neurons in animal brains that are awake, brains of awake animals, have a very wide threshold and temporal variability. So there's, despite the fact that the membrane potential is going to be the same from spike to spike, there's a difference. So there's some other factor going on that influences spiking that can control behavior, because spiking controls behavior downstream, and that's how we move our stuff and speak and everything. So dendritic integration has some X factor, some mysterious, I don't know if it's mysterious, but some other factor that I think could be coming from internally in the microtubules and in this neuron, but also from other neurons through the gap junctions. So dendritic integration that can affect and alter spiking and influence behavior. And I think consciousness may be happening here, and when consciousness kicks in, that's what changes the spiking threshold. So you can have conscious control of your actions. The next objection is to synaptic strengths, mediate memory, Donald Hebb, LTP, the idea that the proteins, usually thought of as postsynaptic, change their sensitivity, and as the sensitivity goes up, threshold goes down, and this synapse becomes more likely to be activated, and that's how learning occurs. However, these proteins are transient. They last hours to days, and memories can last lifetimes. So where is memory stored? That's a good question. Now, you could say, well, that's in the membrane proteins, it just gets replenished. So how does it get replenished? So here's a neuron, and this is actually a dendrite. This is the axon here. And membrane receptors and enzymes and proteins and stuff that need to get transported from the cell body where they're synthesized down to a synapse here or anywhere are transported along microtubules by a process called axoplasmic transport, or dendroplasmic if it's in the dendrite, by motor proteins, kinesin and dynein, which move along a track and carry the membrane proteins to the right place. So remember, these microtubules are interrupted, so they have to kind of jump tracks and change tracks many times, and then you come to a branching point and they have to decide where to go. So which synapse needs it, there or there? Well, how do they know where to go? Especially since the microtubules are disrupted. Well, it turns out that tau, the famous tau protein from Alzheimer's disease, guides the motor proteins and tells them where and when to get off. It's like traffic light. And I'm sorry I don't have the reference from this paper. It was about 2 years ago. And it tells the motor proteins where to get off. So it's the tau protein that's the traffic signal guiding synaptic plasticity. So you could say, well, the intelligence is in the tau. Well, maybe, but the real intelligence is where the tau is bound on the microtubule lattice. So the information seems to be embedded in the microtubule lattice, binding the tau, and then that guides the motor proteins. So it's not all membrane synaptic plasticity. And as you know, in Alzheimer's disease, the tau falls off, and microtubules depolymerize, and you get Alzheimer's disease. You lose memory, cognition, and eventually consciousness. And now everybody talks about the amyloid plaques, where the genes point to, but the amyloid plaques per se don't cause cognitive dysfunction. We had a recent paper with Rudy Tanzi, who is the Alzheimer's expert at Harvard, and he actually discovered most of the genes for the amyloid plaques, but he believes that it's a problem in the neuron and the neurofibrillary tangles. So here's a microtubule, and the tau falls off and the microtubules destabilizes. and falls apart. Now, it could be that the microtubule destabilizes and then the tau falls off. And one of the possibilities is that we found that zinc, Rudy found that beta amyloid sequesters zinc, lowers the zinc levels, and when you lower zinc levels inside the neurons, you destabilize the microtubules. You don't have this strong force, you have a weaker force binding them together. So low zinc by beta amyloid sequestration may mediate Alzheimer's disease, the bottom line being that your microtubules destabilize. If your microtubules fall apart, you lose cognition. Oops. So, where is memory stored? One thing we know about it is from LTP, long-term potentiation, is that when calcium comes in through synaptic activation, calcium activates calcium calmodulin to form a holoenzyme called calcium calmodulin kinase 2, CAMK2. CAMK2 then binds to microtubules and distributes around that whole neuron and neighboring neurons through the dendritic arbors quite rapidly. And CAMK2 can phosphorylate targets. The question is, what is it phosphorylating? Because that would be a source, a site for memory storage. So CAMK2 is a very interesting molecule. Here it is before it gets activated. It's hexagonal, snowflake shaped. Here we're looking at the top of it, and here we're looking at a side view. Then calcium comes in, and calcium causes it to extend these legs, or arms, I'll call them legs. Kinase domains are called, from this association domain, 6 above and 6 below. And each of these legs can phosphorylate a target, and or not. So you can think of it as a bit. So each of these legs is a bit, and the phosphorylation sites are here. They're actually on the upper part of the foot, if you want to call these feet. And somehow that phosphorylates something which would be a good candidate to store memory. Well, what do you think it phosphorylates? So here's all these microtubules. You can't avoid them. We know that CAMK2 binds to microtubules. So the question is, how does CAMK2, activated CAMK2, relate or bind to microtubules? And the answer is perfectly. There's a perfect match. And this we published in this paper, where if you take the CAMK2 and you overlay it on the microtubule lattice, there's two types of lattices. That's the A lattice and that's the B lattice. Either way, because of the play in the legs, they match up perfectly. And the 6 kinase domains can match 6 tubulin dimers in a hexagonal lattice. And each of the 6 can phosphorylate or not phosphorylate. So you have 6 bits of information conveyed from the synapse to the microtubules via CAMK2. And ordered arrays of bits are called bytes. So basically you have a byte of information potentially conveyed from the synapse to the microtubule by CAMK2. So this is what the force field looks like. It shows that they will bind. And here we show the CAMK2 having landed on the microtubule. And here we show how the phosphorylation occurs. There's 2 mechanisms. One is through the C termini, and the other is by unfolding of the kinase domain. So that, and we down to the amino acid level, so that each of the 6 kinase domains can phosphorylate 1 tubulin in a microtubule lattice. And this gives rise to potential for an enormous amount of information. So here we have 3 possible ways to look at this. One is the top 2 are in the A lattice, and the bottom is the B lattice. The top one, we just go by dimers, and the bottom 2, we go by monomers. So you can phosphorylate this one or this one in a monomer, for example. And depending on how you play the game, you either get the possibility for a 6 tubulin neighborhood of 64 possible bits in the top row, something like 500 in the middle row, to 5,000 in this row. So from 1 CAMK2, depending on which of its kinase domains phosphorylate the tubulin. So the potential for information processing from encoding from synaptic input into the microtubule is vast. Now you could say, well, this phosphorylation doesn't last, and then it could be hardwired in a number of ways by post-translational modifications, the C termini, these little hairs that stick out, metal ions, other ligands that could be induced by the phosphorylation, binding of microtubule proteins, or transfer to neurofilaments. So these are ways that the temporary phosphorylation can be encoded and hardwired. So here we have, just to kind of summarize, a CAMK2 landing on the microtubule, leaving this hexagonal pattern and then going on its way. And this could be imparting information, synaptic information. It doesn't have to be all 6. It could be any permutation of it. Well, how would it be read out? So you have information in the microtubule. So what? What's it going to do? Well, one way, as I showed earlier, is that integration in dendrites can modulate axonal firing. There's a couple seats up here if you guys want to come up. Modulating firing, controlling behavior. So it's possible that this integration and information processing in the dendritic microtubules could be controlling axonal firing that way, forming microtubule map scaffolding, neuronal growth, reshaping synapses, synaptic plasticity, motor protein routing, as I showed you before with the tau, storing memory, and consciousness, which we'll come to in a second. So readout, there's plenty of ways that the microtubule information can be read out to influence the higher level stuff. Well, this relates to information processing in microtubules. And as Gautam mentioned, I've been interested in this for many years, since my days in medical school, looking at microtubules and learning about computer matrices at the same time. And I first published in 1982 in the Journal of Theoretical Biology, and then through the 80s about classical information processing. So here's the structure of a microtubule. Each tubulin, each peanut-shaped protein, we say here can be in one of 2 states, black or white. And what those states are is a critical question. We'll come back to that. And so there's about 10 to the 9th tubulins in a neuron, a billion. And they oscillate in the megahertz, 10 to the 7th. So you get potentially 10 to the 9th times 10 to the 7th, 10 to the 16th operations per second per neuron. This is very disconcerting to my AI colleagues because they claim that the entire brain is 10 to the 16th operations per second, something like that. 10 to the 11th neurons, 10,000 synapses, 1,000 per second. They get 10 to the 16th for the whole brain. So they say, well, when we have a computer in silicon that has 10 to the 16th operations per second, we'll have the equivalent of the brain, the singularity, everybody would be wonderful. Give me your money to sign up. Yes, sir.
I heard oscillations at 10 to the 7th hertz, which I guess you'll explain later, and the readouts and encoding when calmodulin sticks onto it. Does that sticking on and off happen at 10 to the 7th hertz
No.
also, or are those different things?
I think those are different time scales. Yeah, but once the memory is in there, it can participate in the ongoing oscillations and upgrading of a cellular automata, which is basically what the early idea was that it's like a cellular automata or molecular automata where each tubulin, by dipole interaction, that's what this model is based on. We've gone to a slightly better model recently, but cellular automata where the dipole of each interacts with its neighbor at discrete time steps, the megahertz being the discrete time steps, and it updates roughly in the millisecond or megahertz range. So 10 to the 16th operations per second in potentially per neuron, which is vast, and as I said, way more than what the singularity types are projecting. So they're not very happy with me about this. But anyway, so I was going around to neural net meetings and AI meetings in the 80s being very annoying to people saying, no, you got to push the goalpost way, way down. And there's all this information going on. And then one day somebody said, well, let's say you're right, wise guy. How would that explain consciousness? How would that explain joy, love, pinkness, redness, whatever? And I didn't know. I was, this is the hard problem. Dave Chalmers subsequently made famous the hard problem, the nature of experience. Well, we have phenomenal experience. But the same person very fortunately suggested I read a book by Roger Penrose called The Emperor's New Mind, which was many things, including a slap in the face to AI, but he argued for a quantum physics mechanism in consciousness, a type of collapse, that this particular collapse mediated by processes in fundamental space-time geometry that I won't go into, caused this collapse in a moment of consciousness, and there was some kind of quantum computing going on in the brain. Quantum computing meaning that you have not just bits of 1 or 0, but quantum superposition bits of 1 and 0 that interact with many other quantum bits or qubits and then collapse to bits, and that's the answer. So I didn't really understand all that. I read the book, and I read Henry's work, and tried to get into this quantum business, which was pretty foreign to me at the time. But it occurred to me that Roger had a mechanism, but he didn't have a structure. He didn't have a good structure for a quantum computer in the brain. And I had a structure, but I didn't have a mechanism. So I wrote to him, and to make a long story short, we teamed up. He liked the idea of microtubules. He liked the Fibonacci geometry inherent in the A lattice and many other things about it. And it was much smaller and closer to the quantum level and could be a quantum computer. So we teamed up and developed our model called Orchestrated Objective Reduction. I'll talk a little bit more about that in a second. And we were attacked even before our first article came out for various reasons. Patricia Churchland and many others tried to kind of pull the rug out before we actually even published. But and we've had many critics over the years and skeptics, but I'm happy to say we're still kicking, and I think our argument is as strong, if not stronger, than ever. But anyway, after hooking up with Roger, we published a number of papers on quantum computing in microtubules, and we're working on another one now for a journal called Physics of Life Reviews that will invite commentaries, and I hope to invite our severest critics and plus a few friends and see what they say. So we'll come back to the quantum business. The last point is that classical Bayesian probability mathematics governs cognition. And Bayesian is probabilities, interactive probabilities. You probably know more about it than I do, or some of you at least do. Bayesian networks express joint probability distributions over many interrelated hypotheses. And this is an idea of how we think, that we have these probabilities and hypotheses, and you kind of move in a random walk from one to another and then come to a conclusion. This is kind of the standard, I think, in cognitive neuroscience. Now, quantum cognition says the mathematics of quantum mathematics is more applicable to what the brain does than classical Bayesian. And it applies the formalism of quantum theory to model cognitive phenomena, memory concepts, conceptual reason, judgment, perception, decision-making. And here are a number of papers. Boosmeyer and Bruza. Actually, Dirk Ertz has been saying this for years. Krennikov, a number of people going way back. But Boosmeyer and Pathos and Bruza in the last few years have really come on strong with this. And they have, for example, they have a BBS Target article in Press, and I have a response, a commentary in the same issue. I think it's probably the next issue. I'm not sure. So, quantum cognition. What's it good for? Well, for example, and this is from Boosmeyer and Pathos's, Boosmeyer and Wang's actually, a lead article in yet another special issue of a journal called Topics in Cognitive Science that'll be all about this, that they point out that psychological conflict, ambiguity, and uncertainty can be viewed as quantum superposition of multiple possible judgments and beliefs. In other words, instead of going this way or that way on some kind of decision tree, you're in superposition, both states. They somehow interact by quantum entanglement and then can collapse to an answer. The second point would be measurement, answering a question. You know, I'm not really sure. You ask me a question, okay, I have to decide, or I just kind of decide on my own. Reaching a decision reduces possibilities to definite states, constructing reality, collapsing the wave function. And there's different ways to look at this, and I'd like to hear Henry's opinion about it later. But the point is that measurement, however you define it, reduces all these quantum superpositions to definite states as your answer. That's a quantum computation. Another point about quantum mathematics is that previous questions influence subsequent answers. So you can ask a question, the famous one in the book is Gore honest and is Clinton honest? And back when, right after they were in office, if you ask, depending on the sequence you ask them, you get different answers. And that holds for a lot of types of questions. This is non-commutativity. So in classical physics, A times B equals B times A. In quantum physics, A times B does not equal B times A. So there's some context, what happened previously influences subsequent. And finally, judgments and choices may deviate from classical logic, suggesting quantum influence, either random or, according to Roger Penrose, non-computable influences embedded in the fine-scale structure of the universe that can influence these in a way that's not random, but might entail some Platonic values or something like that. Okay, so quantum cognition is this mathematical approach, and from Wikipedia they say, well, the field clearly distinguishes itself from the quantum mind, as it is not reliant on the hypothesis that there is something quantum mechanical about the brain. So they're trying to have their cake and eat it too. They're trying to say we're using quantum mathematics, but we don't want to go on that risky limb that there's quantum biology in the brain, because everybody knows the brain's too warm, wet, and noisy to have quantum computing going on. Nonetheless, some of us do feel that the brain has quantum biology, and it has to. And for example, if a classical system would try to simulate a quantum system, you'd have exponential slowdown, as Feynman showed. It would be, you wouldn't be able to do it. It would involve so much computation. And what would be the point of using a classical system in the brain to to simulate a quantum system if you could have the quantum system. So that would be me, something quantum mechanical about the brain. And so Roger and I came out with our theory in the mid-1990s, and here's the basic idea, that inside a dendrite, and these microtubules should be interrupted, each of the tubulins can not only be in bit states of 1 or 0, as we saw in the black and white, but also quantum superposition of both states is a qubit. And the qubits entangle with other qubits in the same neuron and different neurons through gap junctions, reach a threshold that he defined as E equals h over t, that I'll come to in a minute. When that happens, you have a moment of consciousness. We calculated this, for example, for 40 hertz, how many tubulins and superpositions for it to happen every 25 milliseconds, and got reasonable numbers like 20 to 100,000 neurons, for example. There's a couple problems with this model, however. Well, first of all, here we are in 1994 after the first Tucson conference. We're all younger then. There's Roger, there's me, there's Dave Chalmers, Jeff Tollickson, Seamus, Rhett Savage, a few other people. That was a great trip, and we were hiking across the canyon, and we all got delirious, and that's how we came up with our theory. So basically, to skip ahead, the basic idea is that you have a quantum, the gray would be the superpositions. They reach this threshold by E equals h over t, where E is the amount of superposition. It could also be the separation space-time geometry. Then when it reaches this threshold, time t, there's a self-collapse. The quantum superposition terminates, the quantum computing terminates, chooses definite states, shown in number 4, shown schematically here, shown as space-time separation here. The point is this could be happening in all the neurons that are involved in this entanglement, and that's how you select the classical states, which can then go on to trigger the firing, to make memories, to change the synapses, to do what needs to be done for cognition and consciousness. And you would need a lot more microtubules in one by E equals h over t to have, since they're inversely related, to have a short t, for example, 25 milliseconds, so we can have 40 conscious moments per second. Which is much more useful than having one conscious moment per hour, for example. You wouldn't get much done. And a foe or predator who had more conscious moments per time would have the advantage on you. So we suggested that what happens is there's entanglement from one neuron to another microtubule, so that E involves many neurons. In fact, depending on various factors, like 20 to 100,000 neurons might be connected in this way, entangled, and you can have a conscious moment 40 times a second. And
could you unpack E equals h over t a little bit?
Yeah. This is actually the indeterminacy principle, or a derivative of the indeterminacy principle in quantum mechanics. And where E, it's also very much like the, if you use the speed of light for photons, it would be the electromagnetic formula for photons. But it's Planck's constant over 2 pi, and E is the gravitational self-energy. So to have a superposition, and this is a very tricky business, and I'll defer to Henry on this, but how can something be in two places at the same time? It's really a fundamental question. We know that that happens in quantum mechanics. And Roger's idea was that he took general relativity and he said, well, something over here, let's say this particle over here is a curvature of space-time geometry in one direction. The same particle over here is a curvature in the other direction. So a particle over here would be into the screen, a particle in another place would be out of the screen. You actually have a separation in fundamental space-time geometry. This is very much like the multiple worlds hypothesis that says every superposition branches off to form a whole new universe. But Roger's idea was, no, these separations are unstable, and before you get that far, before you get a whole new universe, you have a little bubble or shred in the universe, but it anneals or self-repairs and snaps back or pops back with a moment of consciousness to one or the other. In this case, it's choosing the one into the screen. So it's indeterminacy principle applied to consciousness, basically. For a quantum or a moment of conscious experience. Because the idea is that every time this happens, you have a conscious moment now experience when this happens. But it also chooses classical states in the microtubules which can, run the neuron. So we connect many microtubules and many neurons through gap junctions by entanglement. And I mentioned the gap junctions. And I just want to say that Gautam was telling me last night, well, I saw his talk at Neuroscience about traveling waves, traveling patterns in hippocampus. And you can have gap junctions that, let's say, all these striped ones are entangled because they're connected by open gap junctions. So as this one closes and this one opens, this whole thing can move. So you can have an envelope moving around the brain, something like this. Being is supposed to mean consciousness happening within it. And I think that it's not like that activity in the brain goes to any one area to have consciousness. Consciousness, an envelope can move around the brain wherever it goes. That's where consciousness is. If it's in your sensory cortex, you're having a sensory experience. If it's in your motor or premotor, you're planning or having a motor experience. If it's wherever, that's what's conscious at that moment. Everything else is on autopilot. So you're driving your car to work or your bike and you're thinking about something else. Your envelope is somewhere over there, but then a horn honks or a light changes and it comes back into the cockpit and you become the pilot, a conscious pilot, and take over. So consciousness, the idea, can move around the brain in these envelopes determined by gap junctions, which are in turn controlled by microtubules. And Gautam and other people have shown these traveling patterns moving around the brain. So I'm suggesting it could be something like that, and you should look at gap junctions in the type of system you're studying. And this is from a paper called The Conscious Pilot. Dendritic synchrony moves through the brain to mediate consciousness. And so according to this, E equals h over t. Here's E and here's t. And this can be measured in terms of gravitational self-energy, space-time separation, mass separated from itself, or the number of tubulins or the number of neurons involved. So as I said, you need a lot. So this would be, the top would be relatively normal 40 hertz gamma synchrony, where you're having 40 conscious moments per second. Now in an altered state, we see high gamma up to 80. Meditators, altered states from certain drugs. You have higher frequency and also higher intensity. I think E also indicates the intensity of the conscious experience. So if you're just kind of dozing and you're having only conscious, 4 hertz conscious moments, the intensity is pretty low. But then something arousing happens, and then you go into the 40 or the 80, you're having more conscious moments per second. And we could even have nested gestalts of, say, delta waves enveloping a series of gamma waves. The point is that consciousness is a sequence of discrete moments. The moments, in our case, are proposed to be quantum state reductions by E equals h over t. So our theory, again, was criticized. One objection being, well, the brain's too warm, wet, and noisy. If you try to build a quantum computer in a laboratory, you need absolute zero temperature because any slight vibration, thermal interaction, will disrupt the delicate quantum effect. Therefore, forget about it in the brain because the brain's too warm, wet, and noisy. And we said at the time, well, the brain, biology's had billions of years to figure this out. Give it a break. It probably evolved a mechanism, although we weren't quite sure what it was at the time. Nonetheless, it was a valid objection we took seriously. Fortunately, the last 6 years, there have been a number of findings in biology, quantum biology, showing quantum coherence, significant functional quantum coherence. I mean, everything's quantum if you go down low enough, but we're talking about it at a functional level. And including groups here at UC Berkeley, Brigitte Whaling and Graham Fleming, working on photosynthesis. So it turns out in plants collect photons, you can see that at the top, into a light harvesting complex, transmit the energy in some way to a reaction center, which turns it into food, which we eat. Without this, we wouldn't be here. We wouldn't have it wouldn't be animals, it wouldn't be anything. So, of course, you want this energy to be transmitted at most efficient way possible. Otherwise, you'd be losing most of the energy. So these people, including those here at Berkeley, showed that in this protein, there are these chromophores made of aromatic rings with a metal. I forget which metal is in them. And what they found was the electronic excitations go through all possible paths at the same time. That's how it's so efficient. Otherwise, we wouldn't get the yield we want, and our tomatoes and bananas wouldn't be as good. So the electronic energy is transported through all these pathways at the same time. A quantum superposition, quantum coherence, entanglement, however you want to describe it, occupying all these different pathways to get from here to here. That's the most efficient way. As little as possible is wasted. So maybe the brain figured out a similar mechanism. And that's what we think. Now, and I'll come back to that point. Another problem that was pointed out to us, not very kindly, was the fact that, but they were right in retrospect, that this idea of a protein changing shape so much, like 10% of its volume, would require a lot of energy and would generate a lot of heat. And it didn't make much sense to have a quantum system that's moving so much, because it wouldn't be quantum. It would interact with the environment too much. That was a valid objection. So we changed, we modified, we took these, this is Roger and I more recently in Sweden. I was kind of being his bodyguard. He was being mobbed after a really wonderful talk he gave. And basically, we took it down to the quantum level. In our original, let me go back. In our original model, we have the protein shape being governed. You can't really see this, but this is supposed to be two aromatic rings, like a phenyl ring or a tryptophan ring, an indole ring in aromatic amino acids. And this goes back to Froelich. This was actually Froelich's original idea, in a hydrophobic pocket. And I was very familiar with hydrophobic pockets, non-polar regions within proteins, because that's where anesthetics bind. And the idea is that these are supposed to be electrons. They flip back and forth. They change the shape of the protein. So this was, one hydrophobic pocket with two aromatic rings changing the shape of the protein. Well, it occurred to us that we don't really need to change the shape of the protein at all. All we need is a change of state. And so we went to the aromatic rings as the key, which is also the same thing in the photosynthesis protein. And if you have one aromatic ring, like benzene up top, it can either oscillate between two states in the valence theory, or in at the top, molecular orbital theory, being in superposition. And one of our critics said, you can't use this as a switch because it's always going to be in that molecular orbital theory configuration of superposition. Well, we never said one, we said two, and actually it turns out more. Now, if you have two rings, then they couple by what are called London forces. So if you have two nonpolar neutral electron clouds, you bring them together, the electrons in one repel the electrons in the other, and you form two dipoles. So you have induced dipoles, and then they oscillate back and forth. And I knew about this because this is how anesthetic gases work. Anesthetic gases form their own London forces in these nonpolar environments inside proteins, preventing consciousness and sparing many other things. So we developed the model to be more just looking at the London force dipoles, characterizing the state of a tubulin, which would have some readout mechanism, without involving the big mechanical change. So I mentioned anesthesia, and the idea that the quantum stuff is happening in internal regions that are nonpolar and hydrophobic goes back to my roots studying anesthesia. And this is from the turn of the century, 20th century. Meyer and Overton, separately, Meyer in Germany and Overton in England, were looking at a group of gases that had anesthetic potency. And they were looking for some physical parameter. They were looking at tadpoles rolling over, going belly up, mice falling over, not breathing, a bunch of different animal tests for what they perceived as loss of consciousness, writing reflex, that sort of thing. And so they knew their potency, which is basically called MAC, mean alveolar concentration. And the lower the MAC, the less you need for a given effect, therefore the more potent the anesthetic. And they looked at a whole bunch of different parameters, and they found that the solubility, the potency of the anesthetic correlated over many orders of magnitude with solubility in olive oil, of all things, a benzene-like medium. And this perfectly straight line over many, many orders of magnitude was pretty astounding. Xenon is the inert gas is an anesthetic. Methoxyfluorine is the most potent anesthetic at 0.25%. Think if you think about where benzene or nonpolar olive oil is in the body, you think of the body. Think of a body ground up, looked at in terms of solubility parameters, where as you go this way, you increase polarity, and as you go this way, you increase nonpolarity or decrease polarity. Well, it turns out this little region here of aromatic rings is where anesthetics bind, highly nonpolar. And these areas are shielded from the polar environment, and I think where quantum activities occur in proteins if they are secluded in a nonpolar hydrophobic region, which is where anesthetics act. So most people in the anesthetic mechanism business, okay, so when Myron Overton found that, most people assumed that anesthetics act in lipid regions of membrane. Since most of the 20th century, they hadn't figured out the proteins were doing the dynamics of it, so they're acting in the lipid regions. Then when it was obvious that proteins mediate excitability, ion channels, receptors, they tried to figure out how anesthetics act on membrane proteins, first extrinsically compressing the proteins, but eventually Franks and Lieb in the 1980s figured out that anesthetics act in hydrophobic pockets within proteins. And then the race was on to figure out which receptor was responsible, to which receptor anesthetics bound to cause loss of consciousness. They forgot about the fact that going back 150 years that Claude Bernard had shown that anesthetics act in the cytoplasm by inhibiting protoplasmic streaming, and which is where the microtubules are, of course. And many, many studies over 15, 20 years of looking at anesthetic effects on membrane receptors and membrane dynamics showed no conclusive results at all. They would potentiate excitatory ones and inhibit inhibitory ones. It didn't make any sense. There was no big picture, and they pretty much gave up. But we think, or I think, that anesthetics are acting in tubulin. And the affinity of anesthetics, and this is from this paper here in PLOS One. Travis Craddock and Jack Jasinski are two of my main collaborators. This is showing anesthetic binding sites in tubulin. These are low affinity sites, and these are high affinity sites. Now, the high affinity sites are still a lot lower, less affinity than for a GABA receptor, which is the favorite receptor for, even though it's an inhibitory receptor for anesthetic binding studies, by about 1,000-fold. So there's 1,000-fold less affinity per tubulin compared to GABA. However, there's about 1 million times more tubulins, well, maybe not 1 million, there's 1,000 times more tubulins per neuron than receptors, and there's at least 8 high affinity sites. So at clinically relevant concentrations, there's every tubulin has probably 1 to 2 anesthetic molecules bound to it, regardless of what's happening at the membrane. So my argument within the anesthesia mechanism field is that anesthetics are, and I was kind of intimidated and backed off of this, but I've been thinking about it in recent years and more studies, and they're looking at membrane receptors as more and more futile. So I'm becoming more and more convinced that the primary site of anesthetic action is in tubulin to inhibit consciousness. Okay, so where in tubulin? So this is a picture of the aromatic rings in tubulin. Let's see. So purple is phenylalanine, blue is tryptophan, green is tyrosine, and up there also I've got structures of serotonin, dopamine, and DMT to show you that they're all very similar. DMT and serotonin have this indole ring, the same as tryptophan, and these are very similar to the ring structures that the photosynthesis people have found that have the electronic quantum coherence. And here are the high affinity binding sites for tubulin, and you can see they're very close. It's pretty much hard to miss being close to these aromatic rings. So what we think is now is that it's a topological quantum computer, which means it's not the state of each tubulin that's in superposition, but the pathway. And that within any one tubulin you can have dipoles going back to the London forces pointing this way, or a quantum superposition of both, a dipole qubit. Now anesthetics come along, and by forming their own London forces, they disperse the dipole and take it away. Therefore, no superposition, no collective dipole, no consciousness, no behavior. And a psychedelic, in principle, and there's actually evidence for this, promotes the quantum state by donating electron resonance energy, and you tend to have more of a quantum superposition state. You reach threshold faster and have higher intensity experiences. So that's kind of...
What do you mean by more of a quantum state?
If you go back to the slope of those conscious moments, let me go back. Oops, I'm going forward. So, for example, the top would be normal consciousness. This is kind of drowsy consciousness. So just look at the slopes. That's normal. So if you have more of a quantum state, you're going to have a steeper slope. And you get to a higher E before reaching T. So we're assuming E is proportional to experiential intensity. So you're having more conscious moments per clock time, which would make the outside world maybe appear slower. And each one is more intense than it would under normal circumstances.
So does that mean that there's more of an E?
Yes.
And so what does that mean from a quantum physics perspective, that there's more?
There's more space-time separation. There's more separation of the dipoles, of the tubulins, or whatever the material is being separated from itself. So it reaches threshold faster. It just increases the slope. And there's evidence from the 70s by Kangan, Green, and Snyder, Sol Snyder, the guy who discovered the opioid receptors, where they looked at a series of phenylethylamines, which are psychoactive drugs, to a receptor, and they found that the potency of the phenylethylamines was directly proportional to their ability to donate electron resonance energy to the receptor. So the drugs, I think, enhance the electron resonance, enhance the tendency to go into a quantum state, enhance the transition probability between dipoles, which pushes it more to a quantum superposition state.
So they actually showed that it actually binds to tubulin?
No, this is on some other receptors, but we're trying to do that experiment. Yeah. In fact, DMT is endogenous. It's in all our neurons. It's all over the brain. What it's doing there, who knows? But we're trying to see right now if it binds to tubulin or not. I mean, everybody assumes it's a 5H2A receptor, binds to the membrane, but we don't know that. And there's a lot of reasons to think that there's a lot more to these drugs than just binding to membrane surface receptors, as there are for anesthetics.
And then the tubulin are both in the cell wall and in the cytoplasm?
Only cytoplasm.
Only cytoplasm.
Well, there used to be thoughts that they were in the membrane also, but I haven't seen that lately, so I'm not sure that's true. I look at them as strictly in the cytoplasm. But they could be entangled or in quantum superposition with membrane proteins also. And they could be in direct contact through actin and G receptor, G coupled proteins, et cetera. But I'm thinking of them in the cytoskeleton, where you want a quantum state to be isolated from the environment. So you don't necessarily want it at the membrane surface where it's going to be, bombarded with chaos and noise from the outside. You want to seclude. So inside the neuron, it's secluded, and then within the tubulin, in these hydrophobic nonpolar regions, it's more secluded, therefore protected from decoherence. Where was I? Okay, well, this is sort of the same thing. So here we have this band that would connect to, it's curving around to the tubulin in the next, and therefore spiral up through the microtubule and get this. If this is a quantum channel, it would get macroscopic. It would go the length of the microtubule. So here we see just the two dipole states of superposition, and here's the anesthetized. Very similar to what I just showed. All right, so quantum probability, quantum mathematical cognition. This is Potthos and Busemeyer, and I'm skipping ahead a little bit, but in their Target article, they have this. They had these graphs of emotional states projected as rays in Hilbert space, and I thought it was pretty brave of them to do that. I'd never seen anything like that, and I thought it was great. But again, Hilbert space is an abstract space. So I'm not, I'm a primarily a biologist essentially, and I think in terms of biological structure. And so in my response to them, I use their happy, whether you have a job and being happy or couple and so forth. And this is my interpretation, where these rays in Hilbert space are actually topological qubits or pathways through a microtubule. So this is a microtubule, and each of these are the tubulins with the aromatic rings, and you can see you follow them, and that could follow and become macroscopic if it's a quantum state throughout the length of the microtubule. And this is another type of, I'm sorry I didn't get the reference from this, I got it off the internet, of another type of quantum, topological quantum computer, where it's the pathway that's important rather than the state of individual bits. And in fact, if you go back, so one of the points of this is if one of these tubulins gets knocked out of whack and gets decoherence, you don't lose the whole quantum state because it gets pulled back in by its neighbors. So a topological qubit is much more efficient than an ordinary qubit of a particle. And in fact, a lot of people are in the quantum computing people are looking for topological quantum computers. Kitaev and Friedman and these guys are looking for the right structure. And I think the microtubules may have evolved as topological quantum computers with these lattice trajectories being representing information. Now, if you do this, you lose information capacity because instead of each of these being a bit, the number I was bragging about before of 10 to the 16th would be a lot smaller, but still way huge. So that's not a problem, I don't think.
So we probably should wrap up.
Okay, just about done. Okay, so another way to look at this is Feynman's path integral, and maybe Henry can comment on this. That particle going from point A to point B occupies all possible paths in between, very much like in the photosynthesis where the electron goes through all those chromophores. And if you put it on a lattice, you get what's called the Feynman quantum chessboard, or quantum walks through all possible pathways. And compared to a Bayesian network where you're doing random walks from here to there to here to there, here to there, in a quantum a bit, you'd be taking all the pathways at the same time and then collapsing to the optimal one. So that's the Feynman quantum chessboard, and we can do the same thing in a microtubule and have different pathways and getting patterns. So how would that pattern scale up to the whole brain? And I'll briefly go through this concept of scale invariance or fractal, one of our fractal-like brain processing structures. Marcus Raichle, Dante Chialvo, Bullmore, Van de Velde, all these, a number of people have been talking about this the last 4 or 5 years in electrophysiological terms. If you measure different scales, different time scales and spatial scales, and go down, you get the same pattern repeating. It's like a fractal in some way. Self-similar electrophysiology, holographic maps in hippocampus, fractal eye movements, distributed memory going back many years, small world networks, grid cell hexagons, dendritic arbors, microtubule networks. So what does this mean? So we have, I think, what's basically a geometric, perhaps hexagonal lattice. And if we get to grid cells in the entorhinal cortex, we have this same hexagonal lattice that really is hard to explain, where the animals seem to know where they are in the spatial domain in a grid. And if you look at different layers, you get the same hexagonal pattern at different scales, kind of like a Google map zooming in and out, suggesting some kind of fractal holographic type arrangement of spatial identity, which reminded me of the Library of Babel, which I read, I think, when I was in college, of a vast library of hexagonal rooms, which seemingly meaningless, but every possible ordering. And there's the Library of Babel, which I think there may be some kind of hexagonal fractal process or geometry going on in the brain. So, to conclude, first, I think we need to consider a deeper scale, finer scale, deeper order of information processing in intraneuronal microtubules to explain cognition and consciousness. Mental activities match quantum mathematics more closely than classical Bayesian mathematics. And you could take the argument, well, the brain's classical, but it simulates quantum, but that doesn't really make sense. As I was saying earlier, if the brain walks, swims, and quacks like a quantum duck, it's probably a quantum duck. Topological quantum computation and dendritic somatic microtubules of integrate and fire brain neurons can regulate firings and control behavior. So I think the essential features of consciousness may well be in dendrites and cell bodies, controlling firings. And consciousness and cognition are likely to involve scale invariant fractal-like representations from microtubules on up. The people who are studying this electrophysiologically stop at the neuron, but I think they have to go further down. And finally, neurological and psychiatric therapy should target microtubules. So, for example, postoperative cognitive dysfunction, we have this problem in anesthesia. Occasionally, elderly people who may have Alzheimer's or even if they don't, aren't quite the same after anesthesia for a while. And there's many reasons, the surgery, the stress, so forth, but there seems to be an effective anesthetic. And if they're disassembling microtubules, that can contribute. Alzheimer's would be the same thing. Concussions, which are epidemic in sports right now, there's a recent finding that the microtubules are shattered in neurons in concussion. Of course, if you have a brain injury, you want to promote microtubule activity for synaptic plasticity, for regeneration of neurons or regeneration of synaptic connections, upkeep and maintenance of existing ones and making new ones. And as far as psychiatric therapies, who knows, maybe depression. So one therapy I just want to mention that we're looking at is transcranial ultrasound. Ultrasound is used for imaging virtually any part of the body. You know, babies in the uterus. We use it in anesthesia to look for blood vessels that we want to stick and nerves that we want to block and so forth. But a guy named Jamie Tyler a number of years ago, there's transcranial electrical, transcranial magnetic stimulation, not to mention all the invasive stimulation techniques. He started doing transcranial ultrasound and found behavioral and electrophysiological effects in animals. And, as Andrew tried it, and it got a very interesting effect, actually. When you do it, you don't feel anything for about a minute, and then you kind of get a buzz, to be honest with you. And it was kind of interesting. And so we did a study that's coming out in Brain Stimulation, I think in the next month, of a transcranial ultrasound effects on mental states, a pilot study. We did a very simple study. We're doing a second study, again, with this machine that we wheel around, and we image. You can see the brain, so we know it's getting through the skull and back. And there's a company that we're working with who's making a headset that's going to, you'll be able to walk around with it. We're going to hope to try on people with brain injury, depression, a number of things, Alzheimer's, concussion, so forth. So the bottom line is take care of your tubules and everything else will be okay. Thank you very much. Sean.
Hi, Stuart. Love the talk. Just one or two minor points before I get into the major argument. Duisberg is indeed here, despite repeated attempts to get rid of him. But he has moved on from oncogenes to a more dogmatic emphasis on aneuploidy. It looks like he's correct.
Yeah, I thought he was always an aneuploidy, but he got in trouble for something else.
He did for HIV.
Yeah.
He's probably right about that as well. But the second one is with respect to the Hodgkin-Huxley model. This does not actually commit you to spiking. And in fact, the Hodgkin-Huxley model can support subthreshold oscillations, which means that you can get very, very complex wave superpositions, which indeed people like Patrick Suppies are looking at as ways of how the vastly complicated.
What do you mean by super? You don't mean quantum superpositions?
Not at the moment, classical. But let's move on to the really critical thing I have to say here. I did I published some stuff with Walter Freeman on really this issue of the synchronized gamma and the effects it has, particularly on the background white noise of the brain. And the conclusion we came to is actually that if we look at how synchronized gamma superposes over the background white noise, we come to a conclusion that conscious moments are about 10 per second maximum. This actually fits with the psychological data. And I would also suggest with respect to what you're doing, that the high gamma, there are people like Brad Wojtek here and Ryan Canolti who have some very good ECOG data. And high gamma is not actually just specific to meditators or anything like that. It looks like we all get a lot of high gamma, not just at 80 hertz as you have there, but in the region of hundreds of hertz. Now, this actually brings us to really the question I want to ask. I believe that the Freeman findings are pretty robust, and he has the notion that what really happens in consciousness is...
Now, which findings? There's 10 conscious moments per second, or
that actually what really is going on is that in the case of synchronized gamma, that it superimposes. We've got a classic. We can do this with look at it as a signal processing problem, and actually we find that you mentioned Marcus Reichley, okay? Marcus Reichley's work on dark energy of the brain, okay?
Default mode.
It obtains here. We have about 20% of the total metabolic demand of the organism is actually consumed by the brain from moment to moment. When this superimposition happens, the metabolic demand goes down by 4 orders of magnitude. This is all solid. We have actually published this. And I'm just wondering if you're open to really a kind of different kind of model, instead of focusing on stuff like tubulin. Basically, an argument that says, right, we do know now from photosynthesis that actually at physiological temperatures we can get quantum coherence, which they told you 20 years ago was nonsense, and it turns out you were correct about that. I wonder if you're open to really a model in which we argue that consciousness is something which is, if you like, ubiquitous, and that what actually happens in particular coherent states of the brain it can actually become salient. That there are these 10 times a second or so when a conscious moment can actually exist, but it may be a slightly different mechanism to what you're talking about.
Well, first of all, 10 per second as opposed to 40 per second is no problem. By E equals h over t, t is then equal to 100 milliseconds as opposed to 25. That's number one. Number two, I showed that envelope for the, I think it was delta, but where you have a number of smaller ones. For example, in visual perception, you might see shape, color, motion, meaning, gestalt, collapse. So all of the briefer ones build up to this envelope where you have a gestalt moment. And Nancy Wolf and I published that in 2001. So I don't have a problem with that. Now, if you're talking about consciousness in the universe and getting into a more of a philosophical, even Buddhist type of approach, I don't have a problem with that either. In fact, Roger, I started to tell Henry this before, but it used to be Roger and I talked in terms of avoiding decoherence until you could reach a OR threshold by E equals h over t. And he realized, and in our last paper, and in the paper we're writing now, he says that really what we see as decoherence is really E equals h over t. So if there's a superposition in the environment somewhere, anywhere, out in the air, wherever, it's going to quickly interact with the environment around it, which is random, and it will reach threshold very quickly, but it'll be random and won't have any cognition, it won't have any information. And so that is what builds the world around us, and that these OR moments everywhere. But we distinguish now Orch-OR from OR. Orch meaning that there's some cognition or information that's associated with it, so that it's an orchestrated moment. It has information embedded in it, as opposed to a sort of random, non-informative moment of consciousness that's happening all around us. So it's kind of a Buddhist thing. Yeah.
Thank you for speaking. I really appreciate laying all this out in a pretty coherent argument. Without getting too much into mechanistic detail, I think really what you provided is more of sort of the approach to understanding perhaps the mechanism of consciousness, maybe taking this to a different level. Now in biology we have the notion of emergent property, and so perhaps consciousness is that. And the other thing that comes to mind is getting back to qualia, which is, could this explain qualia, or is it sort of, in a sense, like what we're doing with neuroimaging, where we're sort of mapping out corollaries of conscious experience? So
Right.
could you comment on that?
Right. Well, your first. Okay, the qualia first. Oh, emergence. Yeah, I in a sense, E equals h over t is an emergence. It defines an emergence. The problem I have with people who talk about, and I don't know exactly what you're referring to, but people have been talking about emergence for decades. That you get complex enough and something happens and you have a novel property. Like wetness from water is an emergent property, and there's a million other examples. And so you get enough complicated processing and consciousness happens. Maybe, but I've yet to see a threshold. I've yet to see a testable prediction. It's just, as far as I can tell, a hand-waving argument. You get complicated enough and something happens. Maybe that's true, but it'd be nice to have an equation, nice to have a prediction. We at least have a prediction. If E equals h over t happens, whether it's in an electron or a buckyball or a tubulin, you're going to have a moment of conscious experience. An electron might have a conscious moment, but it would take 10 million years because E is so small and be very, very feeble, not worth waiting for. But we have all this tubulin so that E gets big and T is small. Now, as far as qualia, we wrote a paper in 1996 on the hard problem, which addresses qualia. Qualia being the phenomenal experience, qualitative aspects of any conscious experience. And what we said was that since Roger had put Platonic values embedded in the fine-scale structure of the universe, we said, well, it's logical to think that qualia, or at least the precursors of qualia, redness, maybe joy, grief, or whatever, are particular configurations in underlying space-time geometry. And if there's a red rose over there, the space-time geometry in it has a particular configuration, and I perceive it, and the space-time geometry in my brain adopts that same configuration. So I'm essentially, you could say I'm entangled with it, or maybe it's just reproducing the same configuration. So the qualia occurs in my brain. So we put qualia at the most fundamental level of the universe, the same place where spin, mass, charge, everything comes from. It's what it is down there. Who knows? Strings, quantum geometry, spin networks, twisters. I don't know. And, Roger has some idea about that. Maybe Henry has some ideas. But the point is that we're ascribing qualia to or at least protoqualia, depending on how you define it, to the most basic level of the universe, irreducible, fundamental, giving rise to these properties, just like the same level gives rise to mass, spin, and charge. Yeah.
So it seems, I'm new to this, so it's going to be a very naive question. It seems like you ascribe quite a lot of computational power to even a single neuron.
Yes.
And there must be an observable as a result of the computation, otherwise you wouldn't have an effect. So what's the experiment that allows you to disambiguate your model from anything classical at the level of a single neuron or two?
Well, I don't. I would refer you to a different type of cell, like a paramecium. A paramecium, if you suck it into a capillary tube, it escapes. If you do it again, it escapes faster and faster. It learns. It can find food, mates, have sex, if that's a sign of intelligence. It can do all kinds of things. Slime mold, an amoeba, one cell, sends out these tendrils, much like axons and dendrites. There have been a number of papers in the last couple of years about slime mold, solving equations, escaping mazes, doing all kinds of things. So how to do it for a neuron, I don't know. I'm not. That's a good question. I have to think about it. Maybe somebody else can think of it. But a single cell, I mean, the point is that a neuron is considered a 1 or a 0 in a network, fire or non-fire, and I think that's an insult to neurons.
Well, but your argument with the other cells, the obstacle there, and I'm not, again, the argument there is that those computations are likely doable with protein networks and kinase and phosphorylation, and then the central model, all that stuff, because they're simple enough that you can. So in other words, that you don't necessarily need your model to have a paramecium turn left or right or have chemotaxis.
You don't need quantum, but you need information processing in microtubules. I'm distinguishing between classical information processing. I'm not saying a paramecium is conscious. I'm saying that it's using its microtubules to process information for cognition. It's probably not, it would have to be still, equals h over t. T would have to be fairly long. So it's kind of interesting that the only time they're absolutely still is when they're having sex. So maybe that was the first conscious experience. And that it would certainly promote promulgation of the species.
Certainly the more interesting part of your model is that quantum computing and this collapse of, into a certain state is responsible for some amount of cognition or neuroprocessing.
That's consciousness. But you can have cognition without consciousness.
Right, but my point is it would be interesting to test the quantum part of what would be the experiment to disambiguate that part from...
Okay, so you're giving me the fact that microtubules process information are responsible for non-conscious...
Let's say that.
Thank you.
I think that's...
I'll quit and go home right now. It's a good question. Actually, here's the experiment I would like to... There's this guy in Japan who's done these amazing experiments, but he hasn't published them yet for various reasons. But he takes a single microtubule, puts 4 electrodes on them, uses 2 to stimulate and 2 to record. And normally, they're good insulators. They don't conduct. However, at certain resonant frequencies, they become conductive. In fact, almost arguably quantum coherent between here and here at certain resonant frequencies. The megahertz, for example, the same resonant frequencies we're using, we're trying to use ultrasound to stimulate microtubules. So the experiment would be you stimulate this single microtubule at a certain resonant frequency and get this quantum conductance, superconductive-like effect. You then add anesthesia. You anesthetize the system, and it goes away. You then use a different anesthetic that's twice as potent, and you need half as much to get the same effect. These are called MAC studies in anesthesia. So if you did that for a series of anesthetics and showed a perfect, like the Meyer-Overton correlation with solubility in olive oil, showed that for inhibition and prevention of quantum coherence in microtubules, that would be a testable that's a testable prediction and an experiment that I think can be done and will be done. Yeah.
So it seems that gap junctions play a pretty essential part in a lot of this processing in your model between neurons. But it's my understanding that gap junctions are sort of the exception to the rule as far as synapses go in the brain, if we want to say they're synapses so much. That they're rather rare as opposed to chemical synapses.
Probably about 10% of the number of chemical synapses, but that's still a huge number.
Okay, but it's my understanding further that they're limited to more certain nuclei. When I think of gap junctions, I think of VMES, the mesencephalic nucleus of the trigeminal.
They're all over cortex. The guy Fukuda in Japan has mapped them out in cortex, and they're all over the place. And interneurons are very rich in gap junctions, and they make dual synapses. So an interneuron will have a gap junction connection and an inhibitory GABA connection with the same neuron. So even if they're 10%, even if they're 1%, how many synapses are in their brain? I mean, you got plenty of gap junctions. And the interesting thing is that they seem to be neuron to neuron, not neuron to glia, which we first thought. At least that's the latest. But interneurons have them, and interneurons connect everything. And wherever you look for them, they seem to be there. They're in thalamus. They're all over the place.
Okay, thank you.
Yeah.
This is exciting because in some sense it's very close to a project I worked on a couple decades ago. Certainly in terms of taking on the usual way that people look at things. So I want to see if there's a way to connect what you're doing with what that was. And what that was basically saying neurons aren't simple, they're really complex, they're tiny, incredibly fast computations happening, nonlinear ones inside dendritic trees. It's a sharpening, not an integrating. So in some sense, that's the direction you're going, but you've gone like a 1,000-fold farther by saying it's at the megahertz level rather than the sub-millisecond level, and it's at the atomic level and the quantum level rather than sort of the spine or small.
Well, it's more at the level of the aromatic rings as qubits, but getting there.
So the one thing is that project was plain old membranes. It was membranes and Hodgkin-Huxley and all the rest, and capacitance and all the boring thermal stuff. And I can imagine a connection between the microtubules and, say, microdendrite computations if I knew how it is that the state of one of these things, whether it's clockwise twisting or counterclockwise or up or down, whatever you call it, how that quantum mechanical state affects membrane voltage. Because it's clear that membrane voltage really matters a whole bunch and somehow turns into action potentials. So what's the crossover? How do the microtubules talk to the membrane voltage so that the things that we know about and believe in actually happen?
Well, first of all, as I showed in that paper by Nondorf et al., there's some factor other than membrane potential that influences firing, and only in awake animals as opposed to in vitro or simulation. They showed this variability. It's one of the earlier slides I showed.
Variability, the Hodgkin-Huxley equations themselves are sort of unstable and they have refractory periods and stuff. So I did variability, so I know there's tons of sources of it. So
well, this would be the major source then. What I would argue is that the major source of this variability in terms of triggering spikes is computation going on in the microtubules inside the dendrite. That dendrite, that soma, but also connected to others by gap junction, so you have this collective integration. If you have a whole layer of dendrites that are connected sideways, so you have a sort of network going that way, and then sideways you have these connections, there's this collective integration in the microtubules which influence the firing. So the Nondorf paper shows that there...
That was the part. How do they influence it?
Okay, so you're asking without affecting the membrane or with affecting the membrane? Well,
spike is a membrane voltage shape. So
on the axon side, I'm talking about the dendrite side. So I'm talking this side of the axon hillock or the axon initiation segment. There's computations going on. So a particular output state of a microtubule might, the C-termini tails, for example, stick up, and they're charged, and they can draw ions or attract or repel ions to influence ionic effects at the membrane. It could be something with some other mechanism, kind of like the inverse of a G-coupled protein or something like that. Or it could be that it doesn't go through the membrane, it goes right through the cytoplasm to the axon hillock. I mean, the other thing about the Nondorf study that was interesting was not only is there this variability, but if you look on the axonal side, the spikes are very vertical as opposed to if there were sequential ion channels opening, this ion channel, this ion, so forth, you would get a slanted slope. But they're almost vertical, which means that these ion channels are opening simultaneously, suggesting that there's some kind of, and some people there's quantum coherence among ion channels, so that they're all entangled and opening at the same time. So, but going back to your question, I think that there could be, there's stuff going on. I mean, the microtubules and the dendrites are not disrupted or interrupted from the well, we don't really know how they interact with the microtubules on the axon initiation segment.
Normally I wouldn't want to interrupt, but I want to just say back what you said, or the part I liked about it, which is that normally you'll have a certain number of ion channels opening from whatever synaptic events there were. That's sort of the standard model, and that's done thermally. Each one of those ion channels opens at the same time, sort of as if synaptic events were completely
at the same time?
Just a sec. In the standard model, they open inside an envelope, but the actual opening of individual channels is presumably uncoordinated, like simple models say the actual synaptic events are uncoordinated, so they tend to blur off and be kind of smooth. If you can get those, the same number, so the same net current, if you can get that lined up to cross a threshold, because the computation, the final regular old computation is nonlinear, that threshold crossing, the mere coordination of the same ion channels, if there's some quantum mechanical effect, would in fact produce that kind of effect.
Well, what I was saying about the ion channel would be on the axonal side, that they all open at the same time. So that's why the spike is vertical as opposed to that. What's happening on the other side, I would say there has to be something intrinsically in the cytoskeleton to account for the non-Dorf findings where you alter the variability, or you alter the spiking threshold on a spike-to-spike basis without affecting membrane potential. So I think there's some other factor, some X factor inside the neuron, something to do with the microtubules. Henry.
My question's about causation and consciousness. You talk about consciousness. Part of the time that you're talking, it sounds like the mechanism, the tubular mechanism, is kind of a mechanistic thing controlled by quantum mechanics, and it will somehow lead to the separation, and then an event will occur, and it will be a conscious event. So, insofar as that choice is random, your consciousness would just be an output.
Well, who said it's random?
I'm saying, insofar as it was random, it would just be an output of the mechanical processes that you're talking about. So, are you saying that no, that once the separation, for example, you had the spiral, the happy spiral, and you had the unhappy spiral.
So let's say they're both in superposition, they collapse to one or the other.
Right, exactly. Are you saying that there's some reason that it's going to collapse to the happy spiral rather than the unhappy spiral, for example?
Well, all the normal influence to go into it. For example, in that paper, he talked about having a job or not having a job. If you have a job, generally you tend to be happy. If you don't have a job, you tend to be not happy. On the other hand, you might hate your job, and it might work the other way.
So there'd be a correlation. Okay.
An influence, right. But still, at the moment of collapse, there's some other influence that kicks in. In Penrose's view, it's not random, but it's platonic from the universe somehow, which, let's say, it would be optimistic. It tends to push into happy rather than unhappy.
So that's what you're saying, that the choice that's made here is going to tend to favor your happy spirals. And
It's going to favor what the values embedded in the universe that are determining.
happy, I'm just
Okay.
letting that the value that
Right.
the universe prefers.
Yeah, it's a process in that medium, and it's the medium, in this case space-time geometry, where the qualia are, where the platonic values are, way down to the Planck scale, are influencing one choice or the other.
Because these two structures, the unhappy structure and the happy structure, physically look probably equally complicated. And
Well, they're different angles.
I'm not saying that the universe knows that this one corresponds to something it values, and this other structure corresponds to something that it doesn't value and abhors, or doesn't want to make that.
You're getting pretty philosophical, and I should say, I don't know if you guys know Henry Stapp. He's like belongs in the Mount Olympus of quantum physics, physicists in this area. And your view is more that consciousness causes collapse.
I'm asking your view.
My view is that consciousness is collapse. That consciousness is self-organizing self-reduction by E equals h over t that occurs whenever E equals h over t. And when the collapse occurs, there's some kick, some influence, kind of like a bias at a slot machine or a roulette wheel that's going to push it one way or the other, that's due to fine structure of the universe, which is where the separation is happening.
And you're saying that bias, the universe favors some things over other things, namely the happy job, have a job.
Yeah, and then you could say, well, why did Hitler happen? Maybe people are wired backwards.
You are saying that the universe has a preference.
Yes. Well, has values.
Has values.
Yeah.
Makes a choice, not random, but favors.
Yeah, Roger says that, and I agree with him.
Okay.
Thank you.
Well, it looks like we have many possibilities to collapse in the next 2 days, so feel free to ask and meet with Stuart. Thank you very much.
Thank you all for being here.